Hello, my name is Joshua Ferrar, and I'm a rising senior biochemistry major here at West Virginia University. And I'm an undergraduate researcher in Dr. Brandon Choi's lab, and my project is on the conformational changes of syntaxin 3B in regulating SNARE complex assembly. A little background on syntaxin 3B. Syntaxin 3B regulates SNARE assembly in the retinas. SNARE assembly happens when light is sensed by the photoreceptors in the retina and converted to electrical signals that are processed at the active zone. And this is followed by synaptic vesicles tethering to the ribbon synapses by the SNARE complex consisting of syntaxin 3B, synaptobrevin, and SNAP25. If SNARE complex assembly does not happen properly because diseases such as X-linked retinoschisis that impact the area disrupt communication between photoreceptors and on bipolar cells, causing blindness. Our main goal is to understand how syntaxin 3B controls neurotransmitter release by adopting different conformational states. We hypothesize that syntaxin 3B is predominantly in a closed conformation, unlike SNARE proteins in different parts of the brain. We also hypothesized that a phosphorylation on residue 14 will promote an open conformation, as well as looked at the LE mutation in the linker region, which will allow syntaxin 3B to adopt an open conformation. We also looked at a SNARE chaperone protein, MUNC18, to understand its effects on the conformational changes of syntaxin 3B. How we did this was syntaxin 3B, the phosphomimetic mutant, and the LE mutant were all grown in an antibiotic solution. When the growth was finished, the proteins were purified using nickel affinity through wash processes that finished with a final elution using imidazole. The histine tag and the salt concentration was changed using dialysis overnight in a low salt buffer, so the next day the sample can be run through the high trap Q with increasing salt concentration. The high trap Q allowed us to further isolate the protein and check and confirm we had it with the fractions from the machine. During growth, the ABI tag was biotinylated so the proteins would be immobilized on the microscope slide. The protein was then labeled overnight with Alexa 555 and 647 and run through the G50 column to separate free dye with labeled syntaxin. All of this led us to use this cutting-edge technique of single molecule fluorescent resonance energy transfer, or SMFRET. This technique allows us to analyze the conformation of a single molecule in real time. This happens by exciting the molecules on the slide with light from lasers in a two-laser system with a red and green laser to see the emissions from the donors being from the green laser and acceptors being from the red laser. This allows us to analyze the distance between donor and acceptor, resulting in either high FRET or close interactions, which means it is in a closed conformation, or low or far interactions, which means it is an open conformation. Here in our first result, we looked at syntaxin 1A found in the brain and syntaxin 3B found predominantly in the retina. The cartoon shows the schematic of the experimental setup that we use. In the middle, there are example traces showing high FRET on top with the red on top of the green followed by a photobleaching event. Then the bottom trace shows low FRET with the red below the green followed by photobleaching. On the top right shows a histogram of FRET efficiencies of hundreds of molecules. The population on the left shows the open state, and on the right shows the closed state. We quantified the bar graph by plotting the percent closed population. This clearly shows that syntaxin 3B is predominantly in a closed state. Then we monitored the effect of the phosphomimetic mutation and the LE mutation. The example traces show us that it has low FRET efficiency since the red is lower than the green. The FRET efficiency histogram shows that there's a shift towards a more open conformation being adopted. This is quantified by the bar graph of the closed percent population. Next, we monitored how efficient the syntaxins form ternary SNARE complex, and we added SNAP25 and synaptobrevin in 10 micromolar concentration. What we found was that the phosphomimetic mutations and the LE mutation efficiently created ternary SNARE complex assembly compared to the wild type. And finally, when we looked at the effect of the SNARE chaperone MUNC18, which is known to bind tightly to syntaxin, locking it in a closed conformation. Interestingly, MUNC18 was not able to close the phosphomimetic mutant and the LE mutant. What we concluded from this was that syntaxin 3B, the wild type, is predominantly in a closed conformation, but the phosphomimetic mutation and the LE mutation are predominantly in an open conformation, allowing for ternary SNARE complex assembly to happen, allowing for efficient membrane fusion of synaptic vesicles. I would like to thank Dr. Brandon Choi for giving me the opportunity to work on this project, and I would like to thank Claire Gething for all of the help on it. And thank you for watching.